We've talked about what we're doing in the laboratory, but what's important to many people is translating breakthrough laboratory research into actual clinical therapies. That's one of the more significant challenges facing medicine today, making novel treatments accessible to patients themselves and bridging that gap between the lab and the clinic. That's the mission of our next speaker. Please welcome Dr. Marc Penn.
So in the early 2000s, actually, President Bush's first address to the nation after he was elected was actually about stem cells. It was kind of a cool era where we were focusing on science and thinking about a different world. And I think it really drove a lot of excitement, a lot of expectation of where this field would go and how we would change medicine. And I want to make the point that this reason we were excited about stem cells wasn't because medicine has failed, it's really because medicine has succeeded. The reality is the mortality from having a heart attack has dropped over the last 30 years when we first figured out to open up the artery from well over 10% to now well less than 5%. But patients with heart failure over that same 20, 30-year period have not done better. So people who would have previously passed away from their heart attack are living, and people are living in an era of chronic disease. And we're looking to stem cells to try to restore that function from our successes really of the 80s and the 90s. There have been pockets of success with stem cells. This one example of a clinical trial I ran with a company called Athersys. They have an allogeneic product, and allogeneic cells, meaning those from somebody else being injected into the patient, make a lot of sense in areas of acute injury, when really you don't have time to go and process the patient's own cells. We took these cells, married them to something called the cricket catheter, which that little needle pops out, goes through the artery of the heart where the stent was just placed and injects the cells into the heart that's recently been damaged. And what we saw in this trial was compared to just infusing the patient's own cells down the coronary artery where the heart attack occurred days later, the injection of the cells immediately after stenting and opening that artery actually led to a very nice dose-dependent improvement in the ejection fraction or the squeeze of the heart, almost normalizing heart function. So in fact, when we think about delivery and cells in the right place, we've actually had successes. My laboratories group really started in the early 2000s with the hypothesis that stem cell-based repair of injured tissue in mammals is a natural process, but clinically inefficient, not because we lack stem cells. You've heard from multiple speakers today. All our organs have stem cells. But the molecular signals that tell our body how to repair are dysregulated. What we believe is we as mammals have evolved to scar instead of heal in an attempt to minimize tumor. And we've gone to identify those molecules that actually force our body to repair. Said in a more simple way, we're all snowmen standing in the snow. To the snowman, snow is a stem cell. They're growing more, right? What occurs in mammals is the orchestra, if you will, is seated. Our stem cells are there through our body. What they lack ultimately are the conductors that tell them how to repair and what to do. And our mission really has been to identify these conductors. We ultimately identified something called stromal cell-derived factor 1 in the early 2000s, and really were the first to push the concept of the paracrine repair mechanism and this idea that we have an endogenous stem cell pool within our body that can repair ourselves. SDF1, as you see, is not there in normalcy. It's gone within 7 days of a heart attack. And when we overexpress it in patients who are on average 10 years out from a heart attack, so they have severe heart failure, in a very nice dose-dependent manner, heart function gets better, the squeeze goes up, and compared to placebos who get worse, and the hearts get smaller, which ultimately leads to better cardiac performance. So the challenge is, in 2001, President Bush shows and pushes us in this new era with tremendous excitement, and 18 years later, we have a lot of evidence, but we don't have a lot of proof that this actually works. And then the question is, how do we move this field forward? There's a lot of headwinds against the stem cell field. From a drug point of view, it's obtainable by physicians, frankly, without any specific expertise. So there's a lot of stem cell clinics, which is a challenge. They're safe to administer, thank goodness, so they're doing it, but goodness knows what they may do. And I often say my patients, and I still practice, my patients go somewhere and get something, I can usually figure out where, I rarely can figure out what. And that's a problem. And they're known to exist and desired by patients, so in fact, the market is really satisfying a need. This is just a map from others' work showing all the stem cell clinics that are available throughout the United States and the different cell types that are ultimately available. Adipose tissue alone can be several different types of cells. And again, not only are, in many cases, the physician educated, but so are the patients. When we look at cells and the potency and the potential, we can all get saline, and saline obviously doesn't do very much. We can then get platelet-rich plasma, or PRP, which many physicians sell as a stem cell product that has a lot of exomes and other things, but the reality is not particularly effective. We can get bone marrow cells which age over time and may be depleted of mesenchymal stem cells. Mechanically derived stromal vascular fraction from our fat is a rich source of cells, but when it's mechanically dissociated, the reality is they're still bound up in matrix and not particularly potent. And then there's enzyme-derived stromal vascular fraction, which really frees the cells up, gives us a therapeutic that we can give intravenously and use in multiple therapeutic ways. Other things like the regulatory process has been a challenge. We're focused on treating chronic disease. Patients don't like placebo-controlled trials in chronic disease. I can tell you from personal experience, if you're a heart failure patient and you get placebo and you figure you got placebo, you're leaving that trial and looking for something else. Patients want therapy and they want to feel better. And finally, we have an issue from a biotech point of view. The reality is the biotechs are limited to a specific therapy that they have. They have, almost by definition, a limited understanding of the mechanism of action. Most of us here today have told you, don't really know why these things work, which makes it challenging and how to design clinical trials. And again, we face expensive trials with limited venture capital interest and patient interest, frankly, because of past failures, and again, patients not wanting placebo. So the classic biotech model really is a biotech company goes to the FDA, can take years to get into patients. They hire a contract research organization, which ultimately is quite expensive, that pays the clinical trials, that delivers the data back to the CRO, who eventually, when everything is done, delivers it back to the company. We've developed something called Veloce Therapeutics model, which really is the biotech company exists. We work with offshore, in this case, the National Stem Cell Ethics Committee of the Bahamas, which can take months to get what has been shown to be safe therapeutics approved within the Bahamas. We have a clinic where we can treat patients, and the clinic can actually, in many cases, not all cases, we support many trials, be a source of revenue to actually help pay for the development of the drugs. The contract research organization, which is US-based, still follows every patient and obtains the data. The company can get data in real time, over time, and ultimately do this much faster at a lower cost to bring therapies to patients that patients want, but to do it in a way where data is obtained so that we can truly figure out what works and who it works and how well it ultimately works. So the success in stem cells in our world is bringing the best science with the right cells for the right indication, with the right delivery, which is often forgotten, with expert physicians. So Okeanos is a clinic in Freeport, Bahamas. It's existed for about 4 years. We've run it for about a year. The medical staff is world-renowned physicians. Concierge medical practice follows every patient for 30 days post-treatment. The Bahamian regulatory process is rigorous and open to first-in-man therapies, as I'll show you. We built out a GMP laboratory there so we can process cells as we need to, and we're able to generate data across multiple indications using the same technologies. As you've seen here this morning, many cell types treat many things. If you're going through an FDA process or an EMA process, you need to reapply for every one of those indications. In this case, we can actually collect data across many indications at once. We have a great facility. We have the only cath lab, I was surprised to learn, on Grand Bahama Island. It exists in a regulated jurisdiction, and we believe it is absolutely imperative that these clinics exist in regulated jurisdictions. Patients need to know that, physicians need to know that. Collection of clinical data, combining therapies to improve efficacy when needed, and ultimately a US-based CRO to collect these data. And all to publish, right? We need to share with the world. We treat multiple conditions, orthopedics, autoimmune, neurologic, cardiac, and others. We've treated about almost 400 patients to date, and we treat about 8 to 10 patients a month. We use an enzyme-derived adipose system. The reason we do that is it is a source of cells that can be given safely intravenously, and it's a very potent cells derived from the fat of the patient, put into a fully automated, sterile, single-use system for the patients. The tissue genesis isolator is the system we used. It's highly efficient, again, closed, automated. It isolates the stromal vascular fraction, which isn't a single cell type, but ultimately is a mixture of mesenchymal and endothelial progenitor cells, and they're the patient's own cells. It's been approved in the Bahamas. It's also approved in Japan, the Ukraine, South Korea. And we installed it at Okeanos in 2017. Tissue genesis is also going through FDA pathways, particularly a phase 2 erectile dysfunction study in the US, which is ongoing. One of the things the Bahamians asked us to do, which is, again, something we can do with the facility and the approach we're taking, is classically, Okeanos had used the Cytori system. There are only 2 automated sterile single-use systems in existence. It's Cytori and it's the isolator. And they asked us to compare. You're switching systems, you should compare. We thought that the isolator would be a more efficient system, but no different. Here we see the efficiency, again, done under a Bahamian protocol of 47 million cells per 100 versus 21. But the key point is stromal vascular fraction, enzyme derived, is stromal vascular fraction. The reality is both systems, despite changes in efficiency, have similar numbers of mesenchymal stem cells and similar numbers of endothelial progenitor cells. So we're recognizing a consistency. What's exciting to me is this is the first time this experiment's ever been done, where from the same patient, the fat has been put in 2 systems, and we compare. And again, data needs to drive this field. It always surprises me, patients spend if I was having bypass surgery, I would understand in great detail who was going to open my chest. And patients who go to stem cell clinics know in great detail what stem cells they may get. They rarely know who's going to give it to them, right? And I had a patient recently challenge me, says, stem cells work for the hip, for the knees, but not for the hip. Well, any internist can hit a hip. I'm one of them, right? I can't hit a knee. I can't hit a hip. If you don't have an orthopedic surgeon who's there, you don't know where those stem cells will ultimately go. 95% of clinics who treat heart failure in the United States don't have a cardiologist associated with them. These are problems. We need to have qualified people, and these are just an example of the 3 we have, who are all outstanding, one of whom sitting right there. Stem cell clinics need to be relevant today and tomorrow. We have a very relevant system for today. We want to be relevant for tomorrow. Well, one of the things I've learned, Dracula almost had it right. It's not about the blood, it's really about the stem cells, right? And there's a lot of people looking at anti-aging, and as are we, and advanced regenerative medical technologies has taken advantage of something called parabiosis. So parabiosis is this idea that if you sew 2 mice together, they develop a common blood supply. If one mouse is old and one mouse is young, the older mouse starts to look younger again. There's some passage of something important from the young mouse to the old mouse. Advanced Regen has figured out how to do this parabiosis by actually culturing the young cells with the old cells. So those are the young cells on the top, the old cells on the bottom. The young cells send exosomes, pockets of the cell surface, that contain something called mRNAs, minimally interfering RNAs, that ultimately change the biology or the gene expression of the old cells. When we take those and we inject them into humanized mice that have been humanized with the old patient cells, all of a sudden the immune system wakes up, the lymphocyte numbers go up, the immune system is more active, natural killer cell numbers go up, and we have a phenotype of the marrow, the immune system, that's actually much younger. So this is just what we've done since May of 17 till we've treated the first patient using this approach at Okeanos on December 11th. And in collaboration with the Bahamian regulatory process, we've now treated 3 patients. These are the data from the first patient. What we see 15 weeks later is an increase in the responsiveness to a stimulant, about 27% and 23%, depending on which stimulants you use. But more importantly, when you activate the T cells with an antigen, something that kind of ticks them off, if you will, you have 150 and a 300% increase in T cell responsiveness compared to how the patient was 15 weeks previously. So we can actually now do parabiosis not by sewing people together, but by actually using their stem cells to retrain each other and improve overall function. Now, when we look at the aging profile further, the myeloid to lymphoid ratio, the inflammation versus the immune system cells gets better, goes down. The natural killer cell assay, again, is up significantly. And when we look at gene expression in these cells that are circulating in the bloodstream, the senescence panel, 60 genes that mark senescence, sleepiness of the cells, goes significantly down, and the aging profile of the gene expression goes significantly down, and the cells look more like young cells than they did like old. So incredibly exciting, but ultimately able to be done because we have a rigorous regulatory process that's open to first in man where we can actually treat patients in a relatively quick way. Again, paracrine factors is something my group really launched. The work done by me and Dr. Mayorga, who's in the audience, has now identified CAMKK1 as a kinase that activates the secretome, the paracrine factors that mesenchymal stem cells release. We actually can significantly improve heart function here, seen in the darker red and the darker blue compared to just giving the cells alone. And now, because we understand mechanism, we've actually learned that we can deliver the CAMKK1 remotely. We injected these rats in the thigh and ultimately improved heart function. So now we're starting to get to a direction where we can start regenerative or healing processes in the ambulance by injecting the patient with the gene therapy on the way to the hospital to be treated. And that's really an exciting thing to be able to do. So ultimately, we've put these things together in something called Veloce, and the goal here is to be able to allow companies to interact in a platform way so that we can accelerate the delivery of these therapeutics to patients who are truly seeking them in very important and meaningful ways. And, anchored by Okeanos and Freeport, but also by other Okeanoses that will be developed internationally. So how do we move the field forward? And this we are very passionate about, and the key is we do this together. We do not want to poach patients from doctors and send them back, sometimes broken, sometimes not. We want to collaborate with physicians who want to access stem cell therapy for their patients and get data. No one should be getting stem cell therapy without asking, who's going to collect my data, and is my data going to be meaningful? Because without that, we're not going to move this field forward, and ultimately, medicine is driven by data, and that's what we need. So I want to thank you very much for your attention and happy to answer questions. Thank you.
So you finished on, I think, what is a very key point there, data. You're collecting the data. And I'm not saying that you guys are doing this, but we've had instances where adverse data got buried, didn't get reported. Are you ensuring that all data gets reported so that, in fact, the FDA says, okay, we have confidence in this?
That's right. Hence the reason for the US-based CRO and the fact that the data goes from the physician to the CRO and not to us.
So everything gets
Everything
gets
That's
reported.
Right.
How far along are you now? How many patients?
So we've treated about 60 patients since we took over about a year ago.
This is in some orthopedics?
Yeah, so we're probably about a dozen Parkinson's patients, probably about 24 orthopedic patients. We've just restarted our heart failure program, a little bit of erectile dysfunction urology, and some general autoimmune patients, rheumatoid arthritis, things of that nature. And so, it's been a slow process getting started because putting in place a CRO has been, takes time. Interestingly, IRBs, we went to try and get our registry IRB approved. IRBs say this would be a great thing for a registry. We won't do it for an OUS clinic because they don't like medical tourism, but yet if you try to do it right, they slap you on the hands. So we're working our way through that. It's a bit of a paradox.
So finally, what's the time horizon here for these things being really able to be applied more broadly than in the Bahamas?
So our goal is to pull the data down on an annual basis and look to see where we are and look at how patients are benefiting in each of the different indications. We will only be able to take these data back to the FDA to see if we can then do the clinical trials in the United States. But we'll know two things that typically you don't know. One is you have efficacy, and you'll know the level of efficacy that you may have so you can power your study. And those are two key pieces.
Helps you focus that study.
Exactly right.
Very good. Dr. Marc Penn, thank you.
Pleasure.
Pleasure.